In this section, we're going to take a look at how eukaryotes, like you and I, control our genes or regulate our gene expression. We're dealing with Chapter 18 in the Campbell Biology. We have the 9th or 10th edition. Those are the pages that you can use in order to find them. All right, some big questions here. How are genes turned on and off in eukaryotes? When we talked about bacteria, we learned about operons. But how do multicelled creatures like us regulate our genes? Remember, we have a lot of cells, like trillions of cells to deal with, and well, a lot of those cells are very, very specialized, or what we call differentiated for a certain function. At first, when we have just the egg and the sperm cell, not very specialized, not very differentiated, but as we develop from an embryo to a fetus, we have groups of cells that take on very, very specialized or different tasks from one another. So how does that all happen? Again, remember, prokaryotes are single-celled. They have their bacterial genome as a singular chromosome that's circular and floats in the cytoplasm. Bacteria grow very rapidly, can divide very rapidly, and respond to quickly changing environmental circumstances, such as being able to utilize, let's say, lactose as an energy source that's entered their environment, or the ability to all of a sudden start making tryptophan, an important amino acid, when they run out, using, of course, those operons that we discussed before. But eukaryotes, being multicellular, we have evolved to maintain constant internal conditions while facing those ever rapidly changing external conditions, the situation we call homeostasis. And we grow and develop, and we specialize and coordinate all those trillions of cells to run the whole organism. So how do we control those genes? Well, this is an interesting list here. Here's like a summary of what we're going to talk about. 7 different ways to control gene expression. You can take it all the way down into the microscopic to control at the DNA level, and bring it all the way out to a bigger level to the actual proteins after they're made by degrading them. So we'll talk about those more specifically. Let's start with the DNA level. We'll go small first. So remember how DNA works, that you find it in eukaryotes in the nucleus of the cell. And it's not just the DNA, but it's wrapped around those proteins that are called histones. And remember, there's all those different levels of winding, like winding around spools of thread. Make it tighter and tighter, condensing more and more, till we get to see the actual whole chromosome itself. And I will bring our attention to the word nucleosome when you get the DNA wrapped into that little subunit there called a nucleosome, as that'll come up on our next page, which looks like this, like beads on a string. Okay, so what does DNA wrapping around histone proteins really have to do with gene expression? Well, it's actually quite important. If DNA is wrapped too tightly around those proteins, then our polymerases can't get in there and make mRNA transcripts, and we can't express those genes. Again, if it's too tightly coiled, you're not going to get gene expression. It's like having the gene turned off. You basically block mRNA transcription. Here we can actually see on an electron microscope areas where DNA is very tightly packed, makes for a darker image. That's the heterochromatin areas. And where DNA is less or loosely packed, you get these lighter areas. That's called the euchromatin. In the areas where DNA is less packed, that's where you can actually have DNA expression going on, where you can express those genes by making an mRNA transcript and then turning that into proteins. So here's a way we can control that, or well, at least the cell can control it, is by adding certain functional groups. One of those is called an acetyl group. An acetyl group has carbon, oxygen, another carbon, and then 3 hydrogens. By adding this configuration to the histone proteins, it can loosen that DNA up a little bit, which then allows the polymerases to get in there and make transcripts so that we can express those genes. So histone acetylation is one way to control gene expression at the DNA level. Here's another way, but it's the opposite way, where we can turn off DNA or gene expression. It's called methylation. Remember the functional group called a methyl group? It's a CH3. Usually that's added to the cytosines on DNA. Then it makes that DNA rep tighter, and therefore no transcription will happen. So whenever you find a place on DNA that's heavily methylated, that's an area that is blocking transcription factors, and we're not transcribing those genes. Those genes are not being expressed. In fact, this is actually a way that bar bodies are made in female cells that are going to deactivate one of their X chromosomes. So this is a way to do that by simply adding a methyl or a CH3 group to the cytosines. Another way to regulate gene expression is through transcription initiation. We've talked about promoters before, the regions on DNA, which is a sequence, often a TATA box, TATA box, that the polymerase recognizes and binds to, and other transcription factors as well. That would be other proteins bind there. In fact, this whole diagram probably might remind you a little bit of an operon, but it's still a little bit different in the way that it works. So there's the promoter region, and then the enhancer region. This is usually more distant from the actual gene. So the promoter is here where I'm pointing, and the enhancer is kind of quite a bit ways down the line in front of the promoter. So it's a distant control sequence on DNA, and it needs a binding of an activator protein to get it to work. So this would basically, if it would, the activator would bind with the enhancer, then we can start to initiate transcription. So here's what the model looks like. Here's our promoter, the TATA box, the gene right behind it, but the enhancer, the little yellow squares, that's way out in front of the gene. And then on top of it, these little circular-like structures are the activators. And then when they bind together, you can actually see how the DNA folds back on itself. Notice how our enhancer with the activators are now folding backwards and coming more in line with the promoter until they bind with these other transcription factors over here in the purple. So they will bind with the promoter region, and it becomes this whole transcription initiation complex in order to get RNA polymerase going to make an RNA transcript in order to transcribe and therefore express those genes. Silencer proteins can bind to enhancer sequences and actually block the gene transcription so that this wouldn't happen. In this case, in this picture, in this model here, we're seeing activators bind with the enhancers. So it looks like this. This looks very complex. Don't worry about memorizing all these little parts here. Get kind of the big idea of the whole thing, that all of these proteins work together and make the DNA fold back on itself towards the promoter, thereby activating the RNA polymerase to go ahead and transcribe that coding region. Another level of gene expression is post-transcriptional control. So this means after you've already transcribed the mRNA, well, what can we do with it? Well, that mRNA has DNA's code, and within that code could be actually different versions or alternative ways to read it. And one of my favorite pictures is right here. So remember, with DNA, the actual parts that matter, those are called the exons. Those are the parts that have actual genes that we want to transcribe eventually to make some sort of protein. So those are numbered here, 1, 2, 3, 4, and 5. Then the regions in between are the non-coding regions that are called introns that would get spliced out. Well, it turns out that there's alternative ways to splice the mRNA transcript once we get it. That to make a certain gene, maybe we don't need region number 4 to make that protein. Or over here, maybe we don't need region number 3. So we can get 2 different types of protein from the same mRNA transcript just by splicing it differently. This is called alternative RNA splicing. Just variable ways to process our mRNA transcript or to use DNA's code to make different proteins. Our next level of regulation is mRNA degradation. Like, how long does that mRNA last? The longer it lasts, the more ribosomes can bind to it and the more protein we can make from it. Some mRNAs can last for hours and some last even up to weeks amount of time. Remember the different ways that we process RNA, that we add a specialized 5' guanine cap to it, and then many A's at the end, called a poly-A tail, to help with degradation. Kind of protects it on its long journey from going in the nucleus all the way out to the ribosome. So we can regulate gene expression by how long we allow the RNA to last. We can create enzymes that can degrade it or silence it, even if we like. And in fact, this is newer biology that we're just starting to figure out. There's these small interfering RNAs called siRNAs for short, which are short segments of RNA, only about 21 to 28 bases long, that can bind to mRNA and basically stop it from being read by a ribosome. Basically, it causes gene silencing. So in the picture down here, the long green strands are the mRNAs, and the short green strands are the si, or small interfering RNAs, that we see can bind to our mRNA transcript and therefore block ribosomes from reading it and therefore blocking the making of proteins. It can also cause RNA to be degraded as well, functionally turn off our genes. Like it says, it's kind of a hot, newer topic in biology. A shout out to these gentlemen here for their discoveries with RNA interference. And you'll notice it's not that long ago. Our next level of control is controlling translation itself, where we block the initiation of the translation stage. Certain regulatory proteins might attach to a 5' end of an mRNA transcript, therefore blocking the ribosomal units from attaching there and then stopping translation. And then, of course, the next levels where we actually have the proteins, where we can control it at that level. During protein processing, there could be certain enzymes or proteins that interfere with the making or the processing of the protein by stopping it from getting folded correctly, from cleaving or breaking it apart, adding certain sugar or functional groups that can stop it from working, or maybe even activate it into working properties. And then once a protein is already made and finished, there's other molecules that can tag it for destruction, or like the proteasome, which is a structure that can actually degrade the protein. A shout out to these gentlemen for discovering the ubiquitin, which can degrade proteins. Sorry, my screen is kind of malfunctioning here, so I had to improvise a little bit, but I'm almost done. The proteasome degrading machine, basically almost like a garbage disposal or a cell waste disposer to break down proteins into their amino acid fragments. All right, so there is kind of one of our last slides as kind of a summary of how this all works. The different levels of gene regulation, all the way with messing with transcription at the DNA level, all the way to the big level of messing with proteins, how we can change how we express our genes, change basically our phenotypes. I've left this in the slides for you so that you can practice on your own, looking at the different levels and naming them.